Okay, so this is the second video on muscle physiology, and so we are going to start diving into how a muscle actually contracts. So the first thing we're going to do is review the structure of a sarcomere from last time. So last time when we looked at a sarcomere, we were looking at this picture here. All right, so this is one myofibril. And so then you can see the myofilaments, those proteins, and we have one sarcomere that's from Z line to Z line. So now we're just going to isolate and look at that one sarcomere. Okay. So here we have one sarcomere. All right. So we have our Z line would be here. And then we have our second Z line here. So that's our sarcomere from here to here. All right. And we talked about last time the I band and the A band. All right, so remember the I band is going to be where there's only thin filaments. So that would start here and continue this way, all right, to the next point where those thick filaments start. All right, and the A band is where you have your thick filaments, and then there's some areas of overlapping thin filaments. All right, so that's our A band. And then we would start another I band here, and that would continue. Okay, so we have those, and then don't forget we also had our H zone, which is this segment here in the middle where there's no overlapping thin filaments within that A band. Okay, and so what's going to happen when a muscle contracts? All right, this is called the sliding filament theory. Because these proteins, these actin and the myosin, are filaments, right? Thick and thin filaments. And essentially they're going to slide past each other. Okay, so I'll kind of draw it on here. All right, so the actin are going to be sliding, right, towards that H zone in the middle. Okay, so the actin are the ones that are going to be moving. The myosin stays still, and actually they're responsible for pulling on the actin. Okay, and so what's going to happen is these little, I'm going to zoom in a little bit. All right, so for our myosin, we have our little golf club kind of myosin with those myosin heads, all right, here. And what's going to happen is that myosin head is going to bend upwards and grab a hold of the actin. Okay, and so when it does that, remember it's going to break down ATP and pivot. And as it pivots, it pulls that actin forward. Okay, so that's how that actin is being moved towards the H zone. And all of these myosin heads would be doing that in their own time. So they're all going to be grabbing and pulling and moving that actin forward. So from each side of the H zone, the actin would be moving towards the middle. That's the sliding filament theory. All right, the filaments are sliding past each other. And that is essentially how muscles contract. All right, so two terms associated with that are the cross bridge. All right, so this is when the myosin grabs a hold of actin. All right, so myosin binds to actin. They form a cross bridge, right? So the cross bridge is just the myosin bound to the actin. Let's try that again. Myosin binds actin. All right, so that's the first thing it does. And then remember, it's going to break down ATP, and that will cause it to pivot. All right, so myosin, that is called a power stroke. All right, so myosin pivots after breaking down ATP. Okay, so those two events happen as part of that sliding filament theory. All right. Okay, so that's in general what's happening on the very, very small protein level for muscle contraction. All right, so that's one piece. And so let's talk about what signal causes this contraction to occur. All right, and so we said that's an electrical signal before because these cells are excitable. Muscle cells are excitable. So let's talk about that signal. It's called an action potential. Okay, and so to understand an action potential, we need to understand the idea that excitable cells, really all cells, if a cell is alive, it has a membrane potential. And all that means is that the cell's membrane has a different charge on the inside compared to the outside. All right, so if we're looking at, this is our cell membrane, all right, our extracellular fluid, that's on the outside, cellular fluid here. All right, we have our intracellular fluid on the inside of the cell here. All right, so the inside is going to be slightly more negative than the outside. Okay, and the outside would have a slight positive charge. Okay, and so then with an action potential, what happens is we change the charge. All right. And so it's a change in charge across the membrane. So with this one, right, this is just an example of our membrane potential. So our whole membrane just has this positive charge on the outside, negative charge on the inside. Okay, so let's talk about what happens when you have an action potential. All right, so on a graph, if we have on our y-axis here voltage, all right, generally that's measured in millivolts, okay? And then across the bottom we have time. These would be milliseconds, okay, potentially even smaller, all right? What we're gonna see is that if we start at kind of a negative voltage, all right? So I'll just make up kind of, it's usually maybe around like minus 70 or so. We'll get into the details and the nitty gritty with our nervous system chapter. This is just to understand an action potential. Okay, and so all this negative means is that the inside of the cell is minus 70 millivolts more negative than the outside of the cell. Okay, and so in an action potential, we're going to change that. All right, so that membrane potential, the charge of the membrane, is going to slowly get more and more and more positive. Okay, and the reason that occurs, this is important to understand for muscle contraction, this is the really important part, all right, is if we have our normal membrane potential here, negative and positive, what happens is that channel proteins in the membrane open, all right, and they allow sodium ions. Remember, there's a high concentration of sodium on the outside of the cell. Actually, here, let's write that in up here. We're gonna have a little more space. So we have a lot of sodium, high sodium on the outside of the cell, and our high potassium on the inside of the cell. That was from our sodium potassium pump that we talked about. All right, and so to change the charge of the membrane, you open a channel protein in the membrane that allows sodium to rush into the cell. Okay, sodium has a positive charge, and so adding all those positive charges to the inside of the cell is going to make this charge more positive. Okay, so now we've changed the charge, all right, and made it positive on the inside, negative on the outside, because we've moved all of our positive sodiums from the outside. Now they're in here, okay? And so that's what's happening on our green part of our action potential. So that cell membrane becomes more positive. The charge, the voltage, becomes more and more and more positive, all right? After that, and this is not as important for muscle contraction, right? We'll talk about this a lot more with the nervous system, but after that, the membrane potential will come back down, and that's just because another channel protein for potassium will open that'll allow potassium to go out. So now you have a positive charge leaving, and that'll reset us back to our positive on the outside, negative on the inside. Okay, but essentially, an action potential, that electrical signal, is occurring because you're allowing ions to move across the membrane and change the charge of the membrane. Okay, so that's our action potential, a change in charge of the membrane. Okay, so normally the membrane is negative on the inside, positive on the outside. During an action potential, it becomes positive on the inside and negative on the outside. All right, and that's because in this case we have sodium moving into the cell, making it positive. All right. Then our last piece of the puzzle is going to be, where does this electrical signal come from? Okay, and so it is going to come from the nervous system. All right, and so we have a neuromuscular junction. All right, and so what happens here at this neuromuscular junction is you have a motor neuron. All right, this is a specific type of neuron that communicates with skeletal muscle. It tells skeletal muscle when to contract. Okay, so skeletal muscle cells are not self-excitable, meaning they can't start their own action potentials. So they have to be told by a motor neuron, right, part of the nervous system. This information would be coming from the brain to that motor neuron, and then that motor neuron would send that information to the skeletal muscle, okay, telling the muscle to contract. All right, so the point where that information goes from the neuron to the muscle is the neuromuscular junction. That's where the neuron meets the muscle. Okay, so neuromuscular and a junction where the two things meet. All right, and so the part of the neuron that interacts with the muscle is called the axon terminal. All right, so on this picture, this yellow kind of bulge here, right? So this would be the neuron, and it ends in this axon terminal, which kind of has like a bulb shape. Okay, that's the axon terminal. It's part of the, it's like the ending part of the motor neuron. Okay, all right, so the way that this axon terminal takes an action potential and sends it to the muscle, we're going to see, is going to be through the neurotransmitter acetylcholine. All right, so let's just talk through the process of how this happens. So I'm going to zoom in. All right, so we have an action potential. It's moving through this neuron, right? And it's going to be sent to the muscle to tell it to contract. So we have that change in charge, positive charge on the inside, negative on the outside. That's our action potential. What the action potential is going to do first is it's going to reach this channel protein. This channel protein is called, and we're going to see this a couple times, a voltage gated channel. And so this particular one is a calcium channel. All right, so this is a voltage gated calcium channel. All right. And so what the voltage gated part means, and we're going to see it over and over, is that this is a channel through the membrane, just a tube through the membrane, and it's opened by a change in charge like an action potential. Okay, so voltage gated channels are opened by a change in charge. Okay. All right, so this in particular is a voltage gated calcium channel. All right, so what it's going to do is we're going to have, so, you know, calcium is going to be on the outside of this neuron. All right, and it's going to open and allow that calcium to move in. When that calcium enters, one thing calcium does is it stimulates the release of secretory vesicles. Okay, so it's going to go and tell these, so we have these vesicles that are full of the neurotransmitter acetylcholine. All right, so this is a neurotransmitter. in the axon terminal. Okay, and so it's going to be located in vesicles. All right, just those little bubbles. Okay, so that's what these little green dots inside of here, that would be acetylcholine. Okay, so that acetylcholine in the vesicles, right, and you're going to have calcium coming in, and it's going to trigger those vesicles to, I think black is a little more visible, all right, to move towards the membrane, right, for exocytosis. Okay, so calcium triggers exocytosis of these vesicles containing acetylcholine. All right, and so all exocytosis is, it just means that this vesicle moves closer and closer to the membrane until it fuses with it like this, all right? And then that releases the contents, in this case, the acetylcholine, out of that axon terminal. So that's exocytosis. Okay, and so it's going to release it into this space here. All right, there's a space between the axon terminal and the muscle cell itself. That is called the synaptic cleft. Okay, so the synaptic cleft is the space between the axon terminal and the muscle. Okay, so that would be, if we kind of bracketed this, right, space, that's our synaptic cleft. All right, so that acetylcholine is going to be released into the synaptic cleft. It's going to diffuse out, all right, and what it's going to do is it's going to find acetylcholine receptors, right? So the acetylcholine wants to locate a receptor for itself, acetylcholine. Those are going to be located on the muscle. Okay, and so there's a special part of the muscle. You can see how it gets like all wavy, all right, and it has receptors on it for acetylcholine. That area of the muscle that has these receptors, is called the motor end plate. All right, so this is the part of the muscle. Let's say this part of the muscle cell membrane with acetylcholine receptors. Okay, and so these acetylcholine receptors are a special type of receptor called a chemically, or you may see in your textbook, I can't remember if it says chemically, sometimes they'll say ligand gated. It means the same thing. All right, gated sodium channel. Okay, so we had before our voltage gated calcium channels. All right, so voltage gated means that a change in charge causes the channel to open. Here we have a chemically gated channel, and this just means that when some chemical binds to it, in this case acetylcholine, that will cause the channel to open. All right, so this one opens when a specific chemical, right? They're usually specific for something. This one's a specific for acetylcholine, all right, binds. Okay. All right, so we have our motor end plate here, right? That's on our muscle cell. It has acetylcholine receptors all embedded in its membrane. So that acetylcholine is going to be exocytosed from that synaptic, from that axon terminal. It'll diffuse into the synaptic cleft, and it'll find a receptor. When it binds to that receptor, what's going to happen is it's going to open, and sodium is going to rush into that muscle cell. All right, so that's going to happen at all of these receptors that are binding to acetylcholine. Okay, when it does that, remember, an action potential is caused by the moving in of sodium into the cell, changing the charge of the membrane. right? So now we're changing the charge and we have an action potential, right, in our muscle cell, okay? So the function, we'll zoom out here, all right? So the function of the neuromuscular junction is to take an electrical signal, all right? That's our action potential in the neuron. We want to transfer it to our muscle cell. But electrical signals can't just jump across a gap like the synaptic cleft. So we have to transform it into a chemical signal. All right, that would be our acetylcholine. And then that acetylcholine now initiates our action potential, right? Our electrical signal in our muscle cell. Okay, so that's the purpose of our neuromuscular junction. It's to take the action potential that is in the motor neuron and transfer it to the muscle cell. And so it's transferred by acetylcholine. Okay, so this is the first step to muscle contraction, right? Which is a series of many steps. Okay, so what we're going to do now is we are going to take everything we've talked about so far and put it all together in one story for muscle contraction. And so I think this will help to kind of assemble all the pieces and make sense of it and how they work together. And if you can, in detail, discuss this process we're about to go through from start to finish, then you should be pretty well prepared for this aspect of your exam. All right, so I'm going to be making, all right, so obviously this is hand drawn. I hand drew it and scanned it in so we can fill it in, but I'll make another kind of copy of it and post that online for you guys to, even if it's just, the scanned version of this. And I'll post that online for you guys to use to just practice this process. So if you can fill this in from memory, all right, and I've got it numbered for the different kind of steps, right? 1, 2, 3, 4, 5, and so on. If you can go through all that, then for the process of muscle contraction for your exam, you should be really well prepared. Okay, so let's start with what we just talked about, all right, and then move all the way through the muscle actually contracting. Okay, so we're going to start with number 1. So for number 1, right, an action potential is moving down the membrane of our motor neuron. Okay, it's going to reach a voltage-gated calcium channel. All right. And when that action potential reaches the voltage-gated calcium channel, it causes that channel to open. Calcium is going to move into the axon terminal. All right, that calcium, and this would be our axon terminal here. The moving in of that calcium triggers vesicles containing acetylcholine to be exocytosed. All right, so that's what's happening here. Our vesicle is moving towards the membrane, all right, merges with the membrane. That acetylcholine can then diffuse across our synaptic cleft. Yeah, there we go, cleft. Okay. And what it'll do is it'll diffuse across and it will bind to an acetylcholine receptor. All right. So an ACH receptor. Remember, that is a chemically gated channel. Okay. All right. And those acetylcholine receptors are located on the motor end plate. That's the area of the muscle cell that interacts with that axon terminal, and it contains those acetylcholine receptors. So this would be our motor end plate here. All right. When that acetylcholine binds to the receptor, that receptor will open, sodium will move into our muscle cell, and that will start an action potential in our muscle cell. Okay? All right, so from here, that action potential is going to move down the sarcolemma, all right, of our muscle cell until it reaches these T tubules. It will then move down the T tubules, right, towards the inside of the cell until it reaches our sarcoplasmic reticulum. All right, that's going to be in the orange. Okay, so let's label first our T tubules here, okay, and then our sarcoplasmic reticulum here. Okay. All right. And so when that change in charge, that action potential reaches the sarcoplasmic reticulum, it triggers the calcium stored in the sarcoplasmic reticulum. Remember, that's the job of the sarcoplasmic reticulum, to store calcium. It triggers all this calcium to be released. Okay. There's kind of a more complicated mechanism involved that we're not going to worry about for this class. All right. Just know that this action potential triggers the release of that calcium. All the steps in between you don't have to worry about. Okay. So that calcium has been released, and that calcium is now going to go to our sarcomere. All right. And it is going to bind to that troponin. Okay, so let's label some stuff on our sarcomere. All right, so with our sarcomere, this would be our thick filaments, our myosin. All right, so this is our myosin. Okay, then the blue would be our actin, right? Those little beads. So this is the actin, or the thin filaments. That green line sitting on top of our beads is, remember, the tropomyosin. That tropomyosin sits on the actin and covers up the binding sites on actin for myosin. And then the little spots on top of that tropomyosin that are kind of orange, that's going to be our troponin. Okay. And then we have here our Z line, which marks the start of our sarcomere. All right. Okay, so let's continue our story. So we have calcium, all right, is going to be released from the sarcoplasmic reticulum. It's going to bind to troponin, all right. When calcium binds to troponin, it causes the troponin to change shape. That change in shape pulls the tropomyosin off of those binding sites on actin. All right, so that's triggered by calcium. Okay, so once those binding sites are now exposed, the myosin has the opportunity to interact with that actin because the binding sites are exposed. So what it needs to do is it needs to hydrolyze or break down our ATP into adenosine diphosphate and a free phosphate. And that causes that myosin to kind of sit up. All right, so it's kind of in a, I like to think of it as being now, right, I'll write this here, in an energized state. Okay, because it just broke down ATP and used that that energy to kind of sit up, right? So now it's in an energized state. Sometimes your book will say maybe it's like cocked, right? So it sits up. I like energized because it kind of helps to relate it to the ATP. Okay, so you break down ATP, myosin enters that energized state where it's sitting up. Now, because it's sitting up, it's close enough to the actin to be able to interact with it. So it binds to actin. All right, when it binds to actin, that ADP and phosphate are going to be released. All right, and this release of that ADP and phosphate causes the power stroke. Okay, so that kind of pivoting of that myosin head. All right, and also this here, where we're in that energized state interacting with actin, that's going to be our cross bridge that we mentioned before. Okay. All right, so then the last step in this process is that myosin needs to let go of actin so we can do it all over again. So it can grab the next segment, like the next bead on actin, and move it forward. Okay, and so to let go, ATP has to bind. All right, so myosin releases actin when ATP binds. All right. Okay. And so this process then will repeat. All right. So it's going to release it, and then we can start back over up here. Okay. So as long as calcium is still present, all right, that myosin can break down the ATP, enter that energized state, all right, and then do the power stroke, then release actin again, and keep going over and over and over again, as long as calcium is still present. Okay, so I don't know why it's talking and writing. I always end up adding like extra letters or leaving letters off. So I apologize. I noticed it later on. Okay. So our last kind of thing to talk about for this whole process is how do you stop muscle contraction? All right. So like I said, this process, all right, will keep going as long as calcium and ATP are present. Okay, and so your cells are always going to be making ATP, so the easiest thing is to get rid of that calcium. Okay, and so what happens is the sarcoplasmic reticulum has a calcium pump. All right, so first let's go back to our axon terminal here. All right, so when the with the motor neuron, all right, it only very briefly sends an action potential, and we'll look at some kind of graphs of those action potentials and, what happens when you send them often, but they're very quick. They're milliseconds. And so you get acetylcholine building up, and then you actually need an enzyme to get rid of that acetylcholine so it doesn't keep activating the muscle cell. All right, and so there's an enzyme called acetylcholine esterase. All right. And so the job of acetylcholine esterase is to break down acetylcholine. All right. That way, when the motor neuron here is done sending action potentials, you remove the acetylcholine from the synaptic cleft, so you stop, that action potential being transferred to the muscle cell. Okay, so to stop muscle contraction, that's the first step. The action potential, the motor neuron has to stop sending action potentials. All right, and this acetylcholine esterase will have to remove the acetylcholine from the synaptic cleft by breaking it down. Okay, and so then at the same time, you'll have this calcium pump here. It'll be active and moving calcium from this space out here back into the sarcoplasmic reticulum. All right, and so that will cause the troponin to release that tropomyosin so it can cover the binding sites again. All right, so moves calcium back into that sarcoplasmic reticulum, the SR. All right, okay, so that's the whole process. I'm just going to briefly, all right, put this up on the screen. So that's the whole process with all the blanks filled in and everything. So when you're practicing you can make sure you haven't missed anything. All right. So make sure when you're doing it, you go through each step and you talk about what's happening. So that all the numbers, what's happening at each of these steps. All right. Okay. So 2 more little concepts to talk about before we wrap up this video. So the first one I just think is really interesting, and now you can kind of understand it now that we've talked about the process of muscle contraction, and that is rigor mortis. All right, and so we know rigor mortis occurs after somebody has died. After a certain number of hours, the body becomes very stiff. And so what happens with rigor mortis is that calcium, all right, so the sarcoplasmic reticulum breaks down, right, as the body is decomposing, and calcium is released from the SR. Okay, so that SR breaks down, the sarcoplasmic reticulum breaks down, releasing all this calcium. They go to the troponin, cause that muscle to contract, right? So the body tenses up, okay? And then the next thing that happens is the body doesn't have ATP, right? So the person has passed away. They're no longer making ATP. And we remember that the job of ATP is to release myosin from actin. Okay, so the reason the body is so stiff during rigor mortis is that ATP is not present to release myosin from actin. So it stays, it contracts with that all that calcium, and it stays contracted because it can't let go. So not present to release myosin from actin. Okay, so that's what's happening with rigor mortis. All right. Okay, and so then let's just talk about motor units really quick, since we just talked about our motor neurons and our neuromuscular junction, and then we can talk about these a little more later. All right, so a motor unit is a motor neuron. So this is just the definition, right? So it's a motor neuron and the muscle cells we say, so the word is it innervates. That's just a fancy way of saying what that neuron is talking to, all right? So innervates, talks to, communicates with, all right? And so what you can see in this picture here, each motor neuron, all right, will innervate generally more than one muscle cell, okay? So muscle cells. Good, I just want to make sure I included that. All right, so if we have this motor neuron here, right, in red, it leaves the spinal cord. So this is the spinal cord. It'll go out to the muscle, and it actually will communicate. It will innervate this 1, 2, 3 separate muscle cells, all right? And it varies from one motor neuron to another how many muscle cells they innervate. So it can be a few, or it can be 100 or more, okay? So you can see the purple one is only innervating 2. All right, and so the different sizes of motor units, all right, kind of determines how much control you have over that muscle, right? So a small motor unit would allow for fine muscle control. Okay, so this would be like in your fingers and your hands. Your eyes have very small motor units. And so your brain, your nervous system can activate just very small amounts of muscles to get this fine motor control versus a very large motor unit All right, so large motor units. Would give little fine control, right? So this would be like your big muscles, like your quadriceps and your thigh, or your biceps, that kind of thing. Little fine control, all right? So they're not very good for fine, delicate movement, all right, like small motor units are, okay? And so once, with a motor unit, once that motor neuron sends an action potential, all of the muscle cells that it innervates are going to contract, all right? So keep that in mind. That's how this works, right? So if this motor unit, or this motor neuron, sends out an action potential, all 3 of these muscle cells will contract, always, okay? All right, and so in order for, let's say this is the biceps, right? And so we have, you're lifting maybe a sheet of paper, all right? You're holding a sheet of paper, and, you know, somebody walks up to you and hands you the textbook for this class, just plops it into your hands, all right? How do we go from just carrying that sheet of paper to now carrying a heavy textbook, all right? What happens there is called recruitment. Okay, so what happens with recruitment is that you increase the number of motor units that are active. So now instead of just this motor unit being active, you would activate this one as well as any other ones in that muscle that would help to increase that muscle's capability of holding the weight. All right, so this would increase the number of active motor units. Okay. All right. Good. So then we will pick up with the next video and talk about some ATP requirements, as well as some other concepts associated with muscle activity and that sort of thing.